start with the renal problem set. The way I would like to do it, right? If you guys are, raise your hands, I will, call on you. You could read the question. Even if you tell me the wrong answer, it's okay, because at least, you could tell me your thought process. I'm not looking in terms of, you got it right. Just as long as you can, give me your thought process, that's all I ask. All right. I see Brian got his hand up. Start with question 1.
Which of the following is not a function of the urinary system? That would be A, excretes abundant protein molecules.
Yeah, ideally protein is not something you secreted, right? Remember I was talking about cells and big proteins, especially like albumin. They're not being secreted into your urine, right? And they want to be circulating in your body. So A is the correct answer. Everything else the kidney does, in fact, do. All right, Angel for number 5.
Typical renal blood flow is about blank percent of cardiac output under resting conditions. So I read 2 articles. One of them said 20, and the other one said 25. So I'm going to go with 25 in this one.
25 is correct. So this book has 25. In the lecture, I was using 20%. Why? Because I was explaining to you guys, right? Like, let's say normal cardiac output, right? I was giving you 5 liters. And then I said, what's the renal blood flow? I said about 20%. Why was I using 20%? Is because if I take 20% of 5 liters, then it's about 1 liter, right? So it's kind of makes the numbers easy, right? So about 1 liter of blood goes into your kidneys. Out of that 1 liter, 60% is plasma. The other 40% is cells, right, and your proteins. So the 60% of the plasma, right, that goes into your kidneys, about 20% of this 60%, which is about 600 ml, right, will be about 120 mLs. So every minute, right, 120 mLs is passing through your kidneys. So it was just basically round numbers, but 25 is correct. So 25% is the correct answer. 36, we go with Milan.
Let's hope this works.
Okay.
The process of filtration occurs at the glomerulus, E.
Yeah, so Milan, you're cutting off a little bit. I'm just going to read it. The process of filtration occurs in E. glomerulus. So yes, E is correct. We're going to go with Ian for 42.
The afferent arteriole in the kidney, C, carries blood to the
glomerulus, correct. So let me actually, I think I can show you guys here, right? So let me go find a good picture for you guys. So you guys can see this well here, right? So this here is, right, the glomerulus, the one I'm circling, and the afferent arterial, right, that's written here is what's bringing blood inside, right? And the blood exits through the efferent arterial, and down the line is basically going to go into the renal vein. So afferent, A, right, is the first letter in the alphabet, if you remember it like that. A, right, A in the alphabet. A is what's going to bring the blood to the kidney. E is later in the alphabet. This is what is going to basically move the blood out of the kidney towards the venous side. So remember that. There may be questions on that on the exam. So A, first letter in the alphabet, is what brings the blood flow to the kidney. So that was 42. Next, we're going to go with 54. We have Lindsay for 54.
Substances larger than plants do not pass through the filtration membrane. Is it C, albumin?
Yeah, albumin is a really big protein, basically one of the proteins that maintains our colloid osmotic pressure exerted by the proteins in the bloodstream. So albumin does not leave the filtration membrane. Maybe if you have pathological states, right? When you have a lot of damage there, then yes. But usually it does not. Everything else here, small particles, they may be filtered, right? 59, we have Curtis.
One mechanism the kidney uses to raise systemic blood pressure is to increase secretion of renin, or the well, I don't know how to pronounce that complex.
Juxtaglomerular complex, right? Juxtaglomerular complex, or they call it juxtaglomerular apparatus. It's basically, it's a combination of cells, macula densa and Paul Kisson. Actually, let me show you guys. I think I have a picture here, which is shown here, right? So here we see the juxtaglomerular complex, right? And it's close to the afferent arterial, right? So you see how the blood is coming through here, and this is our complex, which they show with the arrows here, right? Mesangial cells, macula densa cells. There's some cells that are basically forming these operators. And why are they by the afferent arterial? Is because you want to have the pressure sensor where the blood is coming in, right? I want to know exactly what's the pressure, what's the flow, right? And they're basically like stretch receptors, right? They're sensing it. Your kidneys work obviously continuously every second, right? Your kidneys do not take a break. So I want to know what's the pressure, right? What's the flow, right? I want to know what's the sodium content, so that if there's a problem, I can start to release renin. And these juxtaglomerular complex, or operators as they're called, they're measuring this consistently, right? And these cells that make, make this complex, right? So very important. All right, so we go back to the problem set. Next, we have, I think everybody, Akash, would you like to go next?
Yeah.
Or 62? All right.
The amount of filtrate produced by the kidney each minute is called glomerular filtration rate.
Yeah, so glomerular filtration rate, right? We said usually we have 120 mLs per minute, right? That's flowing into the nephron. The other, I was telling this to Ian, right? If you remember, right, GFR, this is basically corresponds to your zones of your adrenal cortex, right? So this was your adrenal gland, and this is the 3 zones here, right? So if I did like 1, 2, and 3, right? So zona glomerulosa would be the release of aldosterone. This will be zone 1. Zona fascicularta, right? You remember what zona fascicularta releases? Cortisol, right? Glucocorticoid. And then zona reticularis is the 3rd zone. What does it release?
Yeah.
The
Androgen.
Yeah, DHEA. When this comes to your testes or your ovaries, right, you're going to make testosterone or estrogen, respectively. So GFR, if you remember that, right, then you should remember your 1, 2, 3 zones. So it's good. All right, number 66. Irene, 66.
In response to increased levels of aldosterone, the kidneys produce. So we have more aldosterone, right?
Mm-hmm.
Okay, so that will act on the sodium and water content.
Yeah.
Which is the reabsorption, correct?
Correct. So more aldosterone, I'm going to reabsorb salt and water into my system, meaning into the surroundings, into the interstitial.
Okay.
So basically the question here is asking what type of urine will that make? So if I reabsorb salt and water from the urine, am I going to get more concentrated urine or less concentrated urine?
More concentrated.
Oh.
So think about it, right? So let me go back.
Am I thinking it backwards?
So let me so let me ask you this. I'm pulling off sodium from the urine, right?
Yeah.
Because I've got to take it into the body. So will the urine have more concentrated sodium or less concentrated sodium in the urine?
Less concentrated.
Less concentrated. So correct. So which out of these options in my urine tells me I have less sodium?
Okay, level concentration. That would be D.
D, very good, right? So urine with lower concentration of sodium. Why?
Okay.
Because aldosterone has
Yeah.
worked on here to reabsorb salt and water, right, into my body. So it makes sense, right? Urine with
Yeah.
low concentration of sodium.
Yeah.
60.
Gotta
Alright.
think it through. Thank you.
Yeah, no problem. We're going to go with Angel for 71.
When the level of ADH decreases, for this one, I'm not really sure, but I'm gonna go with I think it's D.
D, right? D is correct, right? So level of ADH decreases, so it means less ADH. So when you have less ADH, ADH stands for antidiuretic hormone, right? Antidiuresis, right? Less P is made, right? Antidiuresis. So if I have less ADH, more urine is produced, right? And the osmolarity of the urine will decrease. Why? Because ADH is not pulling that water right out of the urine, so there's more urine, right? And osmolarity of the urine decreases, right? Because the solutes are not being pulled off from it, right? So both more urine is produced and osmolarity of urine decreases. Let me show you on the slide. I think I have a good slide for this. Right, so you see this is the distal part, right, of the nephron, the collecting duct where ADH works. It only does
That's
these
correct.
effects. It only does this effects when the ADH is actually coming into right circulation by the posterior pituitary. When the ADH is not there, it's not going to exert this effect. So you see how it shows you ADH and water reabsorption into the interstitium, right? And here, right, what we see is right, there's no ADH. No ADH, so none of this. So if no water reabsorption, nothing of this, the urine is passing. Right, all of this is passing.
Hey, Nicholas,
Yeah.
You need ADH.
I have a hard time understanding osmolarity. That's going from lower concentration to higher concentration, right?
You're talking about here, so normal osmolarity, right? Osmolarity is about 290 milliosmoles in our body, right? So here, what you see, actually, let me see if I have a picture right here. All right, so this was right, coming in with your urine about 300 milliosmoles. You see, the slide got cut off. You see how this says 30, but it's not 30, it's 300 because you see this zero supposed to have been here. When I guess when the slides were made, these numbers got cut off. So you see 300, 600, 900, 1200. Make sense? Right? The
Yes.
zero is here. I know it got cut off. Right? So what happens is you have movement of your urine through this, right? This part of the nephron is water permeable. This part where am I drawing right now, right? So initially you have both water and solutes that can escape. So if I have 300 milliosmoles, right, and I'm coming in here, right, as you go down, it becomes more concentrated. You see 600, 900, 1200. So water will essentially come out to basically dilute this part, and solutes will come out because from 300, right, it wants to go from least concentration, right, from here, it's going to come out. But,
Got it.
But as we move through this part of the nephron, it's no longer water soluble, meaning water cannot escape. So water is in here, right, is on the urine, but this part can still be permeable to these electrolytes. So here, as the urine is moving up, right, you have movement of these ions from the urine that are still here. So these ions move out, and as you go up, right, here we have, again, becoming the urine is going to become less concentrated. Why? Because the water from the urine cannot escape, so this cannot occur, but electrolytes can still move out. So as the electrolytes move out, right, it becomes less osmotically potent, right? Or right, the amount of particles decreases because they're being pumped out, and it becomes less osmolar. So you see here, right, as it comes up here, and this part of the nephron is permeable to water only if you have ADH present, right? So if you have ADH present, then the water can move out. If there's no ADH present, no fluid can move out. So this part, right, both solutes and fluid can move out, this part only solutes can move out, and this part is under the mercy of ADH, right? And what is all this in the interstitium, right? In your nephrons, in the surrounding area, right? There's a lot of solutes that are present as you go down the gradient, right? So there's more solutes, right? That's what exerts this movement, right, of solutes in and out, right, out of the nephron, I should say. Does that answer your question, or are you still not sure?
No, yeah, that answers my question. Thank you very much.
Okay. All right, we were here, right? 71 was D. Next, I think, was Milan for 73.
Antidiuretic hormone would be A.
A, very good. Increases the permeability, right, of the collecting duct, so water, right, by increasing the number of aquaporins, right? So we increase aquaporins. Just, you guys remember this, right? The last part of the nephron is usually water impermeable unless antidiuretic hormone comes in from the posterior pituitary. If the posterior pituitary secretes ADH, that becomes permeable. When will posterior pituitary secrete ADH? For example, when angiotensin 2 comes in, right, and binds on the receptors there, right, you have more ADH production. By the way, we used to have a medication called Vasopressin. It was in place of the first dose of epinephrine for your cardiac arrest, right? When you do your mega code, we give epi as the first drug. But before, we used to give, 20 units, right, of vasopressin. And the way this works is, right, where you guys know, right, it is going to basically reabsorb water, hopefully increase the blood pressure. So that was what we were trying to do, right, vasopressin. So that was 73. By the way, all the hormones, medications, they're usually in units, right? Your insulin is in units, your heparin units, vasopressin units. So next time we're in the lab, I'll show you some of these drugs so you guys can see. 76, we are at up to Ian.
I'm sorry, is there a reason the vasopressin was phased out?
It's still in ACLS. I think it's still there, but they showed, epinephrine exerts. Every time they do studies, to be quite honest with you, all these drugs, most of them have like type 2, 2A, 2B, 2C. So they're not the best thing in cardiac arrest in the sense that they're not what's going to bring your patient back to life or get ROSC. The number one therapy is always defibrillation, early CPR, all of that, drugs and all that. They go back and forth. So one year they have a study that says, this was shown efficacy. Next year they'll find epinephrine was more efficacious. So the drugs, they have the cycles where one is more in favor, some was less in favor. And I also I have a feeling a lot of these companies that make these drugs, they have strong lobbyists who will say like, this is the drug, we got to give this drug. So there's also that, right? So they have incentive to make these studies to say, well, this drug works, right? But all the drugs are not as good as your CPR, defibrillation, intubation, and your early treatments, your basic, I would say, BLS treatments, right? Hopefully that answers that. 76, who I said we're up to, Ian.
Okay. A patient excretes a large volume of very dilute urine on a continuing basis. This is maybe due to the absence of.
You said B, right?
B.
B, yeah. Absence of ADH, correct. So patient excretes large volumes of urine, right? Very dilute, right? No ADH, no reabsorption of water, no aquaporins. So 76 was that. 115, we're going to go with Lindsay.
What does the glomerular complex do in response to decreased filtration rate?
So think of it like this: less blood flow, less pressure in your kidneys. What does your juxtaglomerular operator secrete?
It would increase renin production?
Yeah, B is correct, right? So you said D, right? Sorry.
D is in dog, yeah. Renin.
Yeah. Looking at it. Increases renin production and release. So 115 is D. If you have decreased filtration, right? Less blood flow, less pressure, right? There's better receptors in your juxtaglomerular apparatus. They will release renin. That's only released, right, exactly when you have low blood pressure, low flow. Otherwise, they don't do this. All right. 116, we are going to go with Brian.
Angiotensin 2 has what effect on the CNS? I believe it's B. AG2 triggers CNS responses, including increasing thirst, ADH production, and sympathetic motor tone.
Yeah, all of this is correct, right? So angiotensin 2 is a very potent vasoconstrictor. It's actually more potent than epi, right? And then the other effect it exerts, right, is going to increase thirst. It's going to work on the posterior pituitary to release ADH. So 116B is the correct choice. Next, we're going to go with some fluid electrolytes and your acid-base problems. Next question, let's see who we have here. We have Irene, 410.
Okay. The principal cation in cytoplasm is, that's inside the cell, right?
Mm-hmm. Correct.
Inside. Okay. So bags of.
Exact. Bags of what?
The potassium.
Okay, very good.
That was too
You
easy.
see, you guys remember, right? And the sea of sodium. So very good. B is the correct choice.
Okay.
Excellent. Akash, we're gonna go with 12.
The ions in higher concentration in the extracellular fluids are sodium and. Okay, so I'm not sure about the. Is it potassium or is it A or B? I'm not sure between those two.
Yeah, so it's not going to be potassium because we just said potassium is inside the cells, right? Cells are bags of potassium in the sea of sodium. So here it's actually, it's going to be an anion, right? So the answer here is chloride, right? Why? Because we want to have, gradients, right? So sodium and chloride are principal ions found in extracellular fluid. So potassium is the positive cation, chloride is the negative anion, and this is what's found outside. So this is in high concentration outside. Now 18, we are thinking back to Angel.
18. Which hormone stimulates the thirst mechanism? I think it's ADH.
ADH is correct, right? ADH is correct. And reabsorbs water, right? And makes you thirsty. Very good. Next, we're going to go with Milan for 19.
Which substance plays a role in determining the rate of sodium absorption? B. Aldosterone.
Yeah, very good. So 19 is B. Which substance plays a role in determining sodium reabsorption? It's aldosterone, specifically, right? Acting on the principal cells, mobilizing, right? The principal cells to basically embed with these sodium channels. So Na can flow inside, right? So aldosterone is correct for that. Next one, we're going to go with Ian for 22.
The amount of potassium excreted by the kidneys is regulated mainly by, isn't it also?
Also aldosterone, right? So very good. So in that diagram, right, let me go back to renal lecture. Let me see if I have, yeah, this one is a perfect example, right? So let me just remove some of this stuff so you guys can see. So this is the principal cell, right? And here, right, you guys see aldosterone, right? Aldosterone's coming in. It's going to work on basically the nucleus. It's going to embed these channels. We see potassium channels. We see sodium channels, right? Sodium channels right here. So as this is our luminal fluid. That's basically where the urine is coming. So as the urine is coming, right, it's going to uptake the sodium from it. The sodium is going to come to these sodium-potassium ATPases. Sodium is leaving. Potassium is coming in, and potassium is going to go here. So potassium is going to leave right into the urine, and sodium is going to be reabsorbed. But basically, that's the function of aldosterone: salt water retention and kaliuresis or kaliuria. So 22 we said was B, right? Again, aldosterone is responsible for that. Medication that blocks this action is spironolactone. 23, who do we have? We have Lindsay.
Secretion of potassium into the urine is be increased by aldosterone.
Increased by aldosterone, right? Yes. Right? So we just talked about why that happens, right? Because of these exchangers, sodium potassium ATPases, which use a lot of ATP. That's why we want to be in aerobic metabolism for them to be working. All right, 28. We have Brian.
The blank plays a key role in maintaining acid-base homeostasis by eliminating carbon dioxide. That is the A respiratory system.
Yeah. So, right, carbon. So CO2, right, plus H2O. We're gonna make carbonic acid, and then this will dissociate into hydrogen and bicarb.
Hey, Nikolai.
Yeah.
I thought it was the buffer system.
So what about the buffer system?
I thought that was the one that was acting on regulating acid balances.
Yeah, but here, look what it says, right? Maintained by eliminating carbon dioxide, right? So what I'm saying is, imagine the system is moving this way, right? So eventually, when you combine hydrogen with bicarb, so this aspect of it, this is basically your renal system, right? But towards here, this is your respiratory system. Why is this? Because where's the CO2 going to essentially be removed? Think of it like this. Right? You have a patient who has diabetic ketoacidosis, and they have a lot of acid, right, in their body, secondary to the ketone production, right? And the body's trying to mobilize to basically maintain their pH. So if I want to maintain my pH, I got to get rid of my carbon dioxide, right? Which system is going to do that? The respiratory system initially, right? You're going to breathe faster. You're going to see respiratory alkalosis start to develop as a compensatory response, right? So initially in the balance of this, the carbon dioxide elimination, right? And that's the question here, right? Is primarily through the respiratory system. Does that answer your question or not really?
Right. Yes. So the buffer system is mainly for respiratory acidosis, right?
So the buffer system is basically maintains your pH balance, and we have a couple of buffer systems, right? One system, right? We see that in the blood, right? That's carrying the carbon dioxide, so it's buffering, right? In the sense that it's offloading oxygen in the tissues, picking up carbon dioxide and moving. And the other thing is we have this bicarbonate system, which is found in your kidneys, right? What kidneys do in the setting of acidosis, they're going to move this into the urine, the hydrogen ion, and it's going to move this into the interstitium. So, basically, keep this. They want to keep the bicarb, and they want to get rid of hydrogen. And in the respiratory system, right, the CO2 that was carried, right, by, let's say, red blood cells, there, it's going to get offloaded. So this is how we maintain our acid-base balance.
All right, thank you.
All right. 30, we are... Who was the last person to go?
Brian.
Okay, Brian. Okay, we're up to Irene then for number 30.
A chemical that minimizes changes in the pH of the body fluid by releasing or binding hydrogen ions is called in other words, it can go either way, positive or negative, right? A buffer.
A buffer, yeah. So essentially, if you want to know what a buffer is in the biochemistry sense, right? A buffer is a substance that basically maintains your pH, right? Very closely to the target level. So let's say if I have, let's just say my pH is 7.0, let's just say, right? A good buffer will maintain this pH within 6.0 and 8.0 range, right? Within 1 point.
Yeah.
Right? So that's a buffer system. The point of the buffer is basically to not allow this to become, 12. To not allow this become 4. By the way, you know how they discovered this, the buffer system? So back in the days, they used to do, experiments which will never be ever allowed to be performed now, right? They were doing this on animals. So I think it was a physician, right? It was a doctor, and he said, he took water and he basically dumped acid into it, and he saw the pH decreased, right, significantly. He made the pH 7, right, and then he dumped acid, and the pH quickly dropped to like 2.0 or like 3.0, and then he injected the same acid into a dog, right, and the dog's pH, right, maybe it was 7.3, went down to like 6.9. And it was not the significant drop. And, this doctor was like, how come I put this acid into this water that was the same pH, and all of a sudden the pH became 2 and 3, and I put the same acid into this dog, and the pH decreased very little, right? So they figure like, there has to be a system that maintains acid-base balance. So that was basically acting as a buffer.
Okay.
Right. All right.
I'm sorry, Nick. What was
yeah.
the answer for 28?
28. Respiratory system.
Thank you. I can go into the advanced if you don't mind. I have my hands
Sure.
up for a while.
No problem. Go ahead. Number 32.
So you say 28 was 8, right?
28 was A, yes, respiratory system.
Okay, what's the next one?
Now we're up to, so 28 was A, 30 was B, the buffer, and then we're up to 32. We're up to 32.
All right, so 32 it's the normal pH range for extracellular fluid. It's 7.3 to 7.45.
Yes.
735 to 7.45. Yes.
Yes, 735, 745. Ideally, our blood is usually about 7.4. That's like your mid-range, right? And then we want to maintain this range. We're going to get to some other questions, but basically what buffer system does, it basically keeps your acid-base balance into this form. If you guys, go forward, right? I don't know how much you guys were learning about this in your book, but we can also interpret like arterial blood gases, right? What is, acidosis, alkalosis, is it respiratory, is it metabolic? So basically when we, get someone's blood gases, right, we look at, pH, right? We look at PCO2, we look at bicarb level, right? And we look at PaO2, which is oxygenation status. This doesn't really, tell us about, acid-base balance, but we basically look at, let's say, at pH, we want it to be at 7.4, right? CO2 we already know, right? 35, 45. Bicarb is usually about 22 to 26. PaO2 is about 80 to 100, right? So this is your arterial blood gases, which we collect in the hospital from the arterial side, right? Usually doctor will come in, or a PA, right? And they will collect it through the radial side. It could be in your, side, but usually commonly done, right, on your radial pulse. They'll basically put a needle and they will draw this blood to basically assess their pH status, PCO2 status, right, bicarb status. The goal is basically to know acid-base status. And we do this quite often on patients who are like on a ventilator, who in acidosis, and want to try to basically, correct their acid-base problems. All right.
Hey, Nick,
Yeah.
how come sometimes to check for sepsis, sometimes I'll just do a lactate, a gray tube, and sometimes they will have to go for arterial blood gases. What?
Sometimes it could be a hard stick. The patient, they may not be able to collect it. By the way, you're talking about, sometimes we can also do this on the venous side, right? And kind of see that in a septic patient. So let me actually show you something. I think you may understand this better from this diagram. Where was it? All right. So this is your circulation, right? So in sepsis, what happens is your organs, because there's profound vasodilation, right? Your organs start to extract a lot of oxygen from your venous side, right? So as the blood is moving, right, through your circulation, all these organs are starting to pull off oxygen from your blood. So we also do a mixed venous blood gas. We collect on the venous side, and we see, right, how much the body's extracting. So let's just say if you went from 80% saturation and now you are, you know, on 60% saturation. So this tells me that the body's organs are extracting way more oxygen from your blood, right? From the venous side. So sometimes we will do a venous sample, right, to do that. But lactate we collect. You could collect it from either side. Sometimes maybe what you're referring to a patient may be like a hard stick. It's hard to obtain blood work. They will call a doctor to get arterial work to basically pull from the arterial side. But we could also do it on the venous side for the lactate. That's not a problem. So lactate you could collect on either side.